Hello everyone. Hello to the next episode of TOXstream. I'm very happy to be here. Welcome everyone. My name is Matteo. I'm going to be your host for the next hour or so. As you know already, we are already at our third episode. So very happy to be here and to see so many people still with us. As you know already, but maybe some people don't know us, this show, this table, this microphone, everything is brought to you by AlterTalks Academy. We are a company which focuses on promoting alternative methods to animal testing and science. And we do it in very different way. TOXstream is just one of our many activities. And if you don't know us, I strongly encourage you to go and check out our website, academy.altertalks.be, because I'm sure you can find some activities of your liking. And if you do, and if you are interested, you can also drop us an email there. You can contact us. And we are always interested in getting in touch with people that share our own passions. But today is TOXstream, and you should know already the drill from our past episode. What we do here is engaging with experts on different topics, either on broadly speaking on science or on alternative method animal testing, also called the new approach methodologies. And today we go for this broadly speaking science, and we have a very interesting episode for you. And the way in which you can interact with us is the usual. You can use the chat. Either you are on LinkedIn, YouTube, Twitch, or Facebook. You can write us. I will do my best to try to bring your comment, your question to our expert, and you can simply discuss and say whatever you want on this specific topic. And the topic that we're going to talk today is quite interesting, and it speaks to us specifically in the way in which we intend science and the way in which we do science. Because we talk about the reproducibility crisis, which if you're a scientist, you probably already know what reproducibility is. If you are not, chances are that you heard these terms here or there, especially with the wording crisis on the side. And in a nutshell, this is the basic fundamental way in which we decide scientifically if something is right or is wrong. Because the way in which it works is that when you find something new and cool, what you do is writing a paper, you publish it, you write what you found, but you specifically also write exactly how you found it, so that whoever wants to follow your step can reach the exact same conclusion as you, hopefully. Because sometimes when other people try to reproduce experiments that were published, sometimes they are successful. And in this case, we are a little bit more sure that the original results were right, but sometimes the results are not successful, so they are not reproduced. And in this case, you need to come back a little bit and try to understand if there was something wrong with one of the two papers. But basically, this is the way in which science autocorrects itself. And as you can see from the phrases reproducibility crisis, you can imagine that there is something, or there could be something wrong in this specific process, which is an issue because, you know, we use science to guide our decision on our society. So if what we publish and what we found is not reproducible, we might be a little bit in trouble. But just to give you a few words on context before I introduce the guest, there are different ways in which you can start to address when these problems arise. And I want to start from one specific point that is not usually used in this context, but I always found it quite interesting. And it's the publication of this paper in 2011, which was titled Filling the Future Experimental Evidence for Anomalous Retroactive Influence on Cognition and Affect, which is a very fancy title to say that the author of this paper claimed that people are able to see in the future. Which sounds weird, but it was published. It was published in a prestigious journal in psychology, and it was peer-reviewed, basically meaning that other scientists read this paper, and they had to conclude if this paper was right or wrong from a methodological point of view. And from a methodological point of view, this paper was right. They could not say, okay, what is written here is wrong. And, you know, this caused a little bit of a stir in the field of psychology, because the conclusion that was drawn from this paper was not that people are able to see into the future, luckily, but rather than the way in which science was made at the time, at least in the psychology field, was a little bit weird, and something needed to be done to correct it. And I think that the best reaction ever was from Professor Van Bakers, that he said that reading this paper made him physically unwell, which I think is the best reaction that you can have to any scientific paper ever. But you may be surprised to know that actually 2011 was not the first time in which this type of issue was raised. Actually, the most important paper in this contest was published quite a number of years ago before, in 2005. And is this one, and was published why most published research findings are false. And as you can understand from the title, the Professor Yanidis, that was the author of the paper, claimed that from a methodological point of view, so focusing on the way in which we do science, we can prove that most of what we published is false, because the methods that we use lack way to be confirmed. There is a high rate of non-replication. So if we try to do again the experiment that we published, we ended up with different results. And that's a huge issue. And that was back in 2005. And from then, we spoke a lot in the community, in the scientific community. We tried to understand exactly how big the problem was. But the issue is that we are still lacking a little bit of real data on understanding exactly how big this issue here is. And one interesting data set comes from a survey that the journal Nature did in 2015 or 16. And the survey was on more than 1,500 scientists. And the question was very simple at the beginning. And they simply asked, okay, have you ever had a problem in reproducing an experiment during your career? And as you can see, the vast majority of the scientists said, yes, of course, in the past I had problem producing something that was published already. And more than half of the scientists, pretty much in every different field, they actually said that, truth to be told, I also have issues in reproducing my own research, which if you're not a scientist, I understand that might sound crazy, but if you are a scientist, not so much. And indeed, when nature asked the same scientists, okay, but then do you think that your field has some problem? Well, most of them, actually 73% of them said, no, I still believe that most of what is published in my own field is fine. And this is insane, especially if you look at who said that, especially since, for example, the chemists, they had the highest rate of confidence in their own field, even though their own experience in reproducing experiment was the worst among all. So this is just a way to say that maybe scientists are not the smartest in the bunch. And Nature went also a little bit further and specifically asked to them if they believed if there is or not a reproducibility crisis, especially in the light of this specific reply, and look at the results.
Is there a reproducibility crisis in science? Nature asked 1,500 researchers this question as part of an online survey. Half agreed there's a significant crisis, and many more identified a slight crisis.
So 90% of the scientists said, yes, there is a crisis. I mean, most of them said that there is a significant crisis, and a few of them, 38%, said that there is a slight crisis, which is always an interesting phrase to use because it's something that can rather describe a situation where your living room is on fire, but the chair you're sitting on is still fine. The bottom line is that, yes, there is a crisis, and we have to find a way to solve it. And to understand a little bit better how big this crisis here is, a few projects arose during the time. The first one, maybe the most famous one, was the so-called Psychology Reproducibility Project. Basically, a number of scientists from psychology field, they got together and they said, okay, now we're going to take 100 experiments that were published in very high journal in the last few years, and we're going to try to reproduce them and see how many of them we can actually successfully reproduce. And now, just before introducing our guest, I want to ask you if you know how much you think this specific project was able to reproduce. And to do that, I invite you to go on slido.com, which is a program that we use for this kind of situation where I can ask you a question. So you can go on slido.com, you can insert this specific numeric code, and you will be able to apply to the question that I just asked you. And while you go through that, I think it's time finally to introduce you our guest, which is an expert in reproducibility. He's a professor at the Instituto Bioquímica Médica Leopoldo de Meis at the Universidade de Brazil, and he's also the coordinator of the Brazilian Reproducibility Initiative, which is another project similar to the Psychology Reproducibility Project. And so I'm very, very happy to introduce you, Professor Olavo Amaral.
Hey.
Hello. Ciao, Olavo. How are you?
Fine.
Good? Great.
Yes.
So I wanted to start, maybe while we give people time to reply a little bit at the question. How did you get involved in reproducibility specifically?
I'm originally a medical doctor by training, but my research background is mostly on behavioral neuroscience in rodents. So I used to do mostly neurobiology in memory models, and both in rodents and in computational models until maybe 5 years ago, I would say. I think I got involved in this because I actually started teaching statistics to life sciences grad students in 2011, I think. And I just realized that, I mean, there were bigger problems in the field of reproducibility than those. Like, I mean, we couldn't solve neuroscience if we couldn't solve we couldn't solve neuroscience problems if we couldn't fix science properly before. I think in talking to students and seeing like what is actually like actual practice in real labs, so like I did get them to bring data and bring analysis and whatever, you do realize like how bad the situation can be, and I just realized it was more useful to try to fix science than to try to fix Alzheimer's disease, whatever. I mean, we had to fix science before we can address these other problems. Of course, I mean, we can try to do it simultaneously, but and it was, I think, a good time to get involved. I think the perception that we have a problem in terms of reproducibility has definitely grown a lot in the last 10 years. So I think around like between we mentioned the Daryl Ben's paper in 2011. I think like between 2010 and 2015 was like I think the turning point for actual awareness of this problem. So I think it was a burgeoning field, and like finding a niche in which there was a lot of work to be done. And frankly, there's still a lot of work to be done. At least in the laboratory sciences. I mean, I think some fields like psychology, for example, which you mentioned, have really done a lot in 10 years in terms of address these problems. Of course, they have not solved them all, but I think the people in experimental psychology are very much aware of this these days, and I think especially young researchers. I think in biomedical science and lab science in particular, I think we're still. I don't think we lag behind in perception. I think people realize there's a problem, but I think we're lag behind in data and ways to address some problems. So I think there's still a lot of work to be done if anyone wants to get involved.
No, I understand. Indeed, I mean, it seems that the reproducibility problem might be at the beginning of science. So indeed, we have to find a way to address it, and we can maybe have a look of what people reply to this question just to see. What did they say? Okay, well, it seems that people are slightly more pessimistic than the reality, which is nice. Well, not too far away.
Yeah.
At the end of the day, it was 36%, if I remember correctly, no?
I think between 36 and 47, depending on how you define what's a successful application.
And
there's a responder bias. People who are watching this are probably are likely to be concerned with this. I mean, the Nature survey showed also has a responder bias, right? Like the people who answer those kind of surveys are people who are. I mean, I did answer that one, so I think you have to if you take like actual like a random sample of lab scientists that are not particularly like interested in watching the Reproducibility Crisis webinar, I think they'll get a bit higher. Although people are very variable, I do ask this question like about like what people perceive at their own areas when I start teaching usually, and we get all kinds of answers. I mean, so a lot of people say like 10-20%, others say 80%, and I'm not sure if this is just a matter of perception or a matter of like actually different fields being different, right?
Okay, and maybe well, I can ask you that because the time when this paper was published, I think it was 2015. So I don't know if you were already a little bit in reproducibility, for what I understand. But so were you surprised by these numbers? You were expecting more or less?
I don't think so. I mean, there's worse numbers. In fact, I mean, if you want to go back a little further in terms of lab science, at least in animals, which is more aware of, there's a very scary 2008 paper on like mouse model of ALS, of amyotrophic lateral sclerosis. They get like 70 drugs and they can't reproduce the effect of any of them in a transgenic mouse model. There's There's a spinal cord injury in rodents project by Oslo Stewart from 10 years ago that also tried to represent, I don't think, 12 experiments and got like 2 or 3 right. So I think like these are all smaller scale, right? The psychology one has much more has much more studies. They did 100 studies, and they have a lot of, in terms of open data. I think they're very much the example to be followed. But and there's John Ioannidis' prediction that most published findings are false. That's actually a theoretical model, so I mean there's not empirical data. But I don't think it really surprised people that were working in the field. There have been better numbers in some areas, worse in others, but I think like around half or a little bit less is not completely surprising. I mean, you should take that with a grain of salt. I mean, it doesn't mean that more than half of the papers are wrong. I mean, the replication itself could be could have some kind of problem, or you could just have like interlab variation and like okay, I did this here and did this, and some kind of condition changed, and that explains it. And again, I think a lot of irreproducibility can ultimately be due just to like low statistical power in some studies. So like if you have a borderline statistical power like 50 or 60 or 70%, you expect things not to and you use a statistical significance criteria, you expect stuff not to reproduce, right? So I mean, there have been arguments that, I mean, they have single replications, they're not huge, they're usually like around the size of the original study. So I mean, you could say that like there might be a bunch of those studies that didn't replicate, might be random fluctuation, or might be just heterogeneity between labs. That's probably not all of the problems. I mean, I'm sure pretty much that a lot of them are wrong. I mean, these guys followed up the same people. I mean, the same general group of people. They have many labs which replicated like a sample of studies. They found interesting, like 30-something labs each, and they found that like heterogeneity is not that great. I mean, it doesn't seem to like just variation from one lab to the next, if you try to reproduce things exactly, explains that much of the problem. They do have the social sciences replication project too, which just did like Nature and Science papers. Their numbers are a little bit better. It's like 50-something to 70, 60-something%. But these guys brought their sample sizes up like 5 times the original, so they have like very large replication studies. So they get something between a third and a little less than half of the papers they can replicate.
Okay, so it's much more difficult to really understand where this is coming from. No, I understand. But okay, maybe we can actually go more in the details of, okay, what the hell, why this is happening? Because as you said, probably from the scientific point of view, scientists were like, yeah, okay, I understand. It's normal in a way. But probably this paper was interesting because he said he was able to show to the world outside, okay, maybe there is a problem inside if you pay attention to that. And the first thing that came to my mind, when I saw this paper, I started my PhD in 2014. So, 6 months later, this paper came out and I was like, what the hell? Why am I doing this? And one of the first things that came to mind a lot was like, well, how many of these is due to the fact that someone makes up data? It's plainly falsification of experiment or someone, inventing stuff. If you have some data on that, it would be difficult to find them.
It's difficult to find that out because I mean, I don't think our system, including like the whole peer review structure, is just not meant to detect that, right? I think if you go to like scientists who admit fabricating data, that's a very small percentage, like I don't know, 1%, 2%, 3%, depending on the survey. So I mean, it doesn't seem to be very common. I think people in clinical research have made the argument that if you really look at data carefully, when we actually have open data, you do find evidence of data anomalies in like more than that. That's I mean, that could be just mistakes and not necessarily fraud, right? But there seems to be data that doesn't seem to be real and like maybe more. Still, I mean, that's not the. I think most people in the field agree that that's not the major problem. There's probably some fake data out there, but it doesn't really begin to explain why you don't replicate more than half of studies in one area. It's probably not that frequent, and you don't need to assume fabrication for things not to reproduce. I mean, again, if you go back to the Ioannidis paper, or like to pretty much anybody who tried to model what we call questionable research practice, which is just like a little bit of analysis flexibility, like being able to like throw out an experiment, do it again, and okay, now I use this because this worked, or like I have two variables, but like but yeah, this one didn't work out, so I'll center my story around this one. I mean, those things are enough to generate irreproducibility, and those are very common. I mean, those are like most scientists will admit doing, and not necessarily out of malice. I mean, again, I think wanting to explore your data is just pretty natural. That said, I think people use frequentist statistics, which are very much meant to test a very specific hypothesis. So if you just like to explain how I explore, I analyze this way and this way and that way, and maybe then I'll try to look at this other. You eventually find stuff, right? And
I agree.
0.05 is a very low bar in terms of, so like people use very loose thresholds in statistics in the life sciences, at least in psychology as well. So it's very easy to generate a false positive just out of a little bit of exploration of your data, right?
And maybe before going to that, because I want to speak a little bit about this questionable research practice, we have one question from Amnika, which I understand may be confusing, which is the difference between reproducing something, replicating something, and repeating something. I mean, I would ask you, Olavo, how would you want to define the difference? Because indeed, there is a difference.
People really don't agree on that. So like, I think the people in the field have generally taken the idea of reproducibility like an umbrella term that defines this. That said, if you actually go to like the National Academies of Science have a proposal, I think in 2019 maybe, that actually we should reuse reproducibility for analysis reproducibility alone. Like if you're doing the experiment again and generating new data, you should call it replicability. I think some people have been migrating to replicability. In the last year, so that's the NAS proposal is this from replication should be like replicating something with new data. Reproducibility is I'm able to with your same data get the same results, which some people call analytic reproducibility. That said, a lot of people use reproducibility for everything, and they just distinguish as like methods reproducibility for like being able to reproduce methods, analysis reproducibility. If I have the data, can I reproduce it? Results reproducibility in terms of like, if I do the same experiment again, can I get this? And inferential reproducibility, like, do I have the same conclusions if I do it again? So you have different proposals, but I mean, ultimately, I think reproduce versus replicated versus repeated can actually be pretty much the same thing depending on who you talk to.
Yeah, I understand. And it's still because also the different phases have different issues, because one thing is being able to do the experiment again. Another thing is to find the same results, which in a way one is replicating, the other one is repeating.
Yeah, that's the Steve Goodman, Daniele Fanelli, John Ioannidis proposal. I like that one, which is like methods reproducibility. I can do experiment. Results reproducibility. I get similar results. Of course, what defines similar results is a matter of discussion. You have to put the pressure on that. And then inferential reproducibility is we agree on the conclusions. That's
Okay, and before going a little bit further in that, because I also have something to show you mostly on the cancer reproducibility project, since we are talking about the questionable research practice, which is something that, again, I think that, as you said, a lot of people actually engage in them without really realizing. And these are all these practices that are not ethically fine, let's say, scientifically speaking, but they are not as serious as a misconduct. And I would like again to ask the people that are at home if they are scientists, if they ever engaged in a scientific experiment or not. If you wanna go on Slido, I have the next question, in which I'm gonna try to ask you and trick you in a way, and try to tell me which one of these practice is not considered a questionable research practice, and you will tell me in a moment what you think of that. But related to what Annika was saying, indeed, the difference between reproducibility and replication, I think it was very interesting what came out from the Cancer Reproducibility Project, which was another project from the same consortium, let's say, of the Psychology Reproducibility Project, where what they actually found out is that the issue when we started was, okay, we want to replicate 193 experiments, and let's see what's happening in cancer. And what they ended up was actually being able only to reproduce 50 of this experiment. And reproduce mean doing it again, meaning that they had problem really doing practically the experiment. And so do you think that here is the issue is more that we are lacking the information to redo the experiment, or the way in which we're doing the experiment is wrong?
Yeah, I think there's a lot of problems. I mean, a lot of them they gave up on lack of information. Some they gave up on lack of materials. Some actually have to give up before actually the method doesn't work, right? I mean, you can actually, I mean, you need a certain percentage of cells to light up to analyze, and they never light up. So like your antibody doesn't work. So like, and that's, I mean, science is hard, right? The lab science is not easy. And those guys did try to replicate very complex papers, right? I mean, they did have a set of like very high impact papers from like Cell Science, Nature, like 30, 40, 50 experiments each, sometimes like very high tech ones. So sometimes it's just like methodologically difficult to do things. I mean, it's not surprising that you can't, that some attempts will fail in terms of like, I can't even start to do the experiment. That said, some of them might be fixable when you're actually lacking information, or lacking because the authors don't help you, or because the materials are not available anywhere in the world because they did them themselves and they didn't share. Those are obstacles that could be addressed, right? So I think the point is like much of this could be solved if people did a better job in reporting, if people did a better job in sharing data and materials.
But I guess it's one of the issues, because one of the things that they said in the paper, and I guess it's related to the question that we just got, is that a decent amount of researchers, they did not collaborate with this project. They didn't apply, basically, when they were asked, can you help us? And
the author response is very variable. I mean, they describe some people as extremely helpful, and some people were just like not helpful at all. I mean, someone even answer. I mean, that happened a lot to us in our project. We can get to that later, but like sometimes you just don't get an answer. Sometimes people say, no, I won't help. And I think it shouldn't help, because I mean, if it's a replication project, you might as well. I mean, if I help, I'm not. It's not really how this should be, and I think that's a valid point. But some people were extremely helpful in terms of like sending materials. So I mean, some authors refuse to collaborate, yes, but not all. So I think there's a big difference in author responses, right?
And follow that, I think there is another question on saying if the fact that a big portion of this paper were not reproduced did somehow ended up changing the way in which the journals that published this paper behaved.
I think the results are fairly recent, so I mean, I don't think I can really talk about the impact on journals. I think the results came out late last year, like December maybe. So I think we still have to see how things turn out. I mean, there have been some effort in the part of journals in trying to work with checklists or some other way, like or like requiring data. So I mean, I think there has been movement in the journal side. That said, I mean, there's a limit to what journals can do in terms of like journals like are really coming in at the end of the process, right? I think there's an inherent problem with the way peer review in academic science normally works, is that there's no control of the process. I mean, you just get the results at the end, and like you're actually like, even if reviewers are doing their perfect job, they're still looking at a very filtered report by the authors, which is they have all kinds of bias over their own results. And so it's like a very summarized and very biased report of like a very complex thing that happened in the past, which we don't have access to. So like I do feel that peer review is like vastly overrated in terms of like what really can achieve, even if like reviewers and journals were doing a perfect job, and they're not. But even if they did, I mean, quality control has to happen during the process, right? It's very hard for journals to fix something that's wrong from the beginning, right?
Yeah, no, I agree. And it's true that the peer review, I mean, when you are a reviewer, you just receive the paper, and maybe you have to read through dozens of pages of RNA-seq data, which are impossible to understand if you're not an expert. And it's indeed an issue. But there is another aspect on that. Maybe
it's not clear what a reviewer should do. Like we've been very keen in like, okay, people should share data, share code, etc. But I mean, it's not absolutely clear that a reviewer should actually check the code. I've had code checked by reviewers, which was fine. They did find errors, which was nice.
Okay.
But like, I'm not sure this is actually expected. It's very unclear what a reviewer should be checking actually, and I think that's another problem, right? I mean, peer review is this abstract concept that okay, you take and some people just like think, okay, I should take a look at the paper, and it's not my job to actually review the fine grain of the data. Some people think otherwise, but I mean, if you don't clarify what a peer reviewer is actually doing, what a peer review is a very generic name. I don't think it's going to work either, right? I think we should like have very specific tasks in terms of like checking this and this and this for things.
Yeah, especially because some of the biggest paper right now, they have so much interdisciplinarity that we cannot probably expect to have just 3 reviewers, which might not be expert in everything.
And maybe not the same reviewer should be checking everything, right? I mean, there's a lot of like very technical statistics issue, etc., that actually could actually be done better by a specialized reviewer that actually looked at just this. So like I'm the data checker here, I'm the code checker, and like and maybe that guy doesn't have to be an expert in the area, right? I mean, to actually like check code, I mean, it's and see if it's reproducible. You can get something with someone like a vastly different expertise, and maybe you should just like, I mean, maybe you just need the expert in the area to actually take a look at the whole thing and see if the conclusion makes sense, etc. I mean, that's very area specific, but like for lots of the just like quality control issues, they might be done better by a professional in quality control.
No, makes sense. So basically, you imagine more methodology specific reviewer, which indeed is right now is more everything. We're gonna look globally at the paper if it makes sense, but rather look at this experiment.
You can atomize it in different portions, right? Like I think more people should be checking different aspects of stuff, and I think it should probably extend it in time. I mean, not everybody should be checking at the end. You should have people actually doing stuff in the beginning, right?
Okay, that would be a huge change in science, but indeed it's an interesting idea. And maybe related to that, there is one video that I want to show you, and you tell me after if this could be also an issue, how much it is an issue on science and reproducibility.
In 2011, a team of physicists reported a startling discovery. Neutrinos traveled faster than the speed of light by 60 billionths of a second in their 730-kilometer trip from Geneva to a detector in Italy. Despite 6 months of double checking, the bizarre discovery refused to yield. But rather than celebrating a physics revolution, the researchers published a cautious paper arguing for continued research in an effort to explain the observed anomaly. In time, the error was tracked to a single incorrectly connected fiber optic cable.
Now, what I really like of this story is, as you were saying, science is hard, and you may screwing up in ways that you don't understand. And there is also a portion of that to the story. It's also the fact that maybe the science that we do is so complicated sometimes that inevitably we ended up with issues like that.
Yeah, I mean, that's how physics is like another universe in terms of like they have very specific. I mean, they have much stronger theory than we do in biology, right? So like they have very strong predictions in terms of like what should happen, what shouldn't happen, very precise predictions of like what I should expect from this. So like if a major anomaly comes up, they do have solid enough theory to say, okay, yeah, we trust. We should trust. I mean, we should keep trusting the theory and like making. I mean, it's probably the experiment that goes wrong. I think they have less of a strong theory tradition. I mean, we don't have theories in biology, but they're very loose and kind of like verbal in nature and not quantitatively precise. So it's like it's hard to sometimes actually mapping what a theory actually predicts in terms of experimental results is actually not as clear. So like you might have this major overarching theory which is like vague enough that like if I get this result, I can fit it into the theory. If I get the other result, I can also fit it. So I think this thing is like, okay, we should not trust the results first, work better when theory is stronger. And I think there's a lot of people have made this case in psychology as well. Like we don't have good enough theory to more than a problem of reproducibility, we have a problem of theory. I think we have both, and it's not like you have to put one against the other. And of course, like to build stronger theories, you need more robust facts. I mean, it goes both ways, right? I mean, if there's a lot of noise in the literature, it's very and you don't really know what to trust and what not to trust. It's very hard to actually move theory forward, because it doesn't challenge you. I mean, if you know that like half of the results is wrong, if this doesn't fit my theory, just say, yeah, this is probably a false positive, and you go on. So like you actually need more robust science to inform theory, and you need more robust theory to help you detect your errors, which was what happened in this case, right?
Yeah, no, I understand. So basically, you're saying that maybe in biomedical sciences we are still more disconnected. We don't have this overarching theory to collect us together and help us going in one direction, or understanding where we're going. Rather,
we do have theories, but they kind of like rise organically, and they like inevitably any theory in biology is like contradicted by like a lot of results, because like there's just like a lot of noise there. So like I think if we just could be more sure about like what facts we're really sure about, because we do know some stuff, right? It's not like it's yeah. I mean, nobody would argue that if you give like insulin to a rat or a human, your glucose levels fall. I mean, that's been known for 100 years, and you don't really need it. I mean, you can. I mean, so like we do know some things for sure, right? But it's if you are, especially if you're not an expert in the field, it's very hard to tell what is really known for sure and is agreed by everybody, and what is actually something that is there in the literature. Some people talk about it, but like a lot of people within the field say, like, yeah, I can't really reproduce this. So like, I think, and it takes a very long while for this facts that like come up in the literature by one big group and like maybe are confirmed by 2 or 3 others out of bias. Eventually, like if it's not true, it probably fade away in like 50 years, but it takes a long time, right? So like I think one of the problems, like peer review is a very, like being published in a peer-reviewed article is actually a very low bar. Like there's, I mean, pretty much anything passes it, especially like with the amount of journals and the heterogeneity in peer review. So like for an outsider, it's very hard to tell what's really known for sure, what is like insulin lowers glucose, and what is just like I don't know, 5G technology creates the coronavirus, which was also published in a peer-reviewed journal formally. So like so I think we need like ways to grade our evidence better. I think clinical research does that a little bit better. Like people in medicine have like systematic ways to assess existing evidence and like grade it and give it like a B, C, whatever rating. I think we lack those, and I think for an outsider, it's very hard just like to look into the field's literature and say like what really can be trusted. This is like consensus. What is not, and it's and if you're like just a newcomer to the field, like a young student, sometimes it takes you a very long time to realize that like a lot of the literature is just not right. And again, like to know what you can trust and you cannot is like you need a lifetime studying a particular field to actually get oriented, which is not very efficient, right?
No, it makes sense. I understand. And maybe because I want to jump as well a little bit on the indeed how we should try to move research in a more clinical or validation setting. But just to finish on the methodology, because you briefly touch upon that, the fact that we can play with the data as much as we want and something might come up. And I'd like to go a little bit deeper in that, because especially on the fact that to evaluate a research or not, we use this so-called significance. So basically we use statistical method, and at some point we say, okay, this thing makes sense and is a rather arbitrary way to do it. And we use this p-value setting that is a way to tell us if what we found is by chance or not. And how much is an issue there? What's the issue there with the way in which we use p-values? What's the way in which we play with this data to make them say what we want?
I think there's 2 issues, or maybe 3, if you want. Like one thing that people really don't understand what a p-value means. Like most biomedical sciences will not be able to give you a correct p-value. Most do. I mean, so it's like it's not the probability that something occurred by chance, but it's actually the probability by chance you might get a similar result if there's no result at all, right? So like people are very keen in inverting the probability and actually like making a p-value sound like, okay, this is the probability, this was by chance, so like the complementary, like if this is 5%, that is 95% this is true. That's not right. I mean, people do they reverse a conditional probability and put up a Bayesian interpretation of p-value. Shouldn't do that. So like one is plain misinterpretation. Second one, again, is flexibility in analysis. So like frequentist statistics are very much about classic statistical tests do test very specific hypotheses, right? And you have to specify it from the start. And if you try to play around, like I can measure this by this and this and this, I can try different statistical tests, or I can just like try it in different variables, or maybe like reform like. Or maybe I look at the data. If I know this might be interesting to test, so like I look at the data and generate a hypothesis from the data and test. So like, it's very easy to get out of the framework. Like I have a clearly specified hypothesis, and I'm doing this, which is the situation in which p-values actually work. If you're actually looking at the data and trying to analyze it in different ways and checking the results, okay, maybe then it just doesn't make sense. I mean, the number doesn't mean anything anymore. And the third thing is that like p of less than 0.05 is a very low threshold. It's a very low threshold, right? I mean, it's, you get a false positive once every 20 out of every 20 experiments in which there's no effects at all. So like if you go to like other fields like physics uses 5 sigma for important stuff, right? So it's like p of 0.0000, I think 7.3 or something like that. I mean, you could just get more robust in terms of like being more using lower alpha thresholds, right?
So I'm feeling very bad compared to physics now.
I think some other like genomics have done interesting, like this big genomic consortia. When they started to actually applying the Bonferroni correction to the whole number of genes you're looking at, they use like a p of 10 to the -7, or something like that. So like you can raise the bar, but like raising the bar actually means doing larger experiments, spending more money, more time. So it's harder to do more robust science. But and again, maybe we don't have to do it always. Like for some stuff, you should be exploring, and actually you're gonna do like a bunch of experiments afterwards to confirm this. I think it's perfectly okay to use like a higher, like a p of 0.05 or even more, like as a first best thing. If again you're going to confirm, like I think there's not. I think we're bad at distinguishing situations in which we're actually like just exploring something and trying to come up with something new. And if we're like less strict, that's fine. And actually doing like now we have to really confirm this is true. So like we need the higher standards, right? We should do both things. But I think we're not very good at differentiating this. And like I think most science is actually done in a very exploratory manner. So I think we do need to develop more confirmatory frameworks. Maybe not for everything, but like for situations where like we have to really agree whether this exists or not.
Well, it's good that you said that, because I think if there are PhD students listening, the first thing that you said, I mean, if you have to have 0.000001 of p-values, they would say, okay, I can quit my PhD. I would never get there. That's cool. And this is, in a way, is also related to also the issue that we have with the bias, with the fact that we only publish positive results. So we tend to publish only positive results, right? We don't really like to publish what doesn't work.
Yeah, that is, yeah. I mean, there's a lot of work on publication bias. I mean, that definitely. I mean, this is not absolute, right? I mean, you do find negative results time and again. I think people would disagree on whether this is getting better or worse. I think it varies a lot in different fields. But yes, that said, I mean, if you take the average biomedical, I mean, it's not like one result, right? It's like 10 or 15 experiments in a paper, right? Or 30 sometimes. So I think there's a bias there's a bias in storytelling as well, which is not like, it's not like everything is positive, but like what I want, like I need the experiments to fit my story. So people are very selective on what they include in terms of like throwing away the experiments is very normal, which is I think the problem. Like it's not necessarily because it's negative, but it's just, I mean, it doesn't agree with my story. Sometimes you need the negative results as a negative control or something, so you want to, so it goes the other way around. But I think it's just a liberty of like you end up writing the paper with a curation of the experiments you want to put in there, and that's a problem, of course, because then you can, I mean, if you do enough experiments, eventually they come out the way you want. That said, I mean, I think the hard part here is that, I mean, sometimes experiments just go wrong because they go wrong. I mean, I can't read this. Like the cells didn't light up. My Western blot is blank. So like this is a methodological failure, right? So like there's no information to be drawn from it, and of course you don't want to publish those because
I agree.
are non-informative, right? But it's so like, but this kind of experiments actually like go into the same bucket as the ones which actually worked perfectly fine, gave a negative result. I mean, these are all like experiments that went wrong, and people are very free into like throwing them in the same bucket, and that's a problem. And of course, if the result doesn't really come out the way you expected, it's very easy to find an excuse and say, oh, yeah, maybe that day the rest were a little bit depressed. Yeah, and there was noise in the experiment room, and if you actually look at this is a little bit weird. The control, so like it's very easy to find an excuse which seems honest in terms of like there is, but it's really just hindsight bias, right? Like now that I know that this is negative, I'll find a way to, and the people do it innocently. I mean, I don't think much of this is actually like done out of malice, but it's once you have the results in, and you can see the results before you say, oh, well, this is valid, this is not. It's very hard to really be unbiased in that sense.
No, I agree. And indeed, we can actually go back to what the people said on the questionable practical research. And you see that, well, majority of people said none, which is right. All of them are in a way or in another questionable research practice. But a lot of people said it's fine to report unexpected finding as having been predicted from the start. Because I guess we all love a good story in a way or in another. It is the way in which our brain works.
But really, I think the problem is the second part of the sentence. Like reporting unexpected findings is fine, and reporting this and okay, I think this sounds really exciting. It's fine too, as long as you're clear that okay, this wasn't my hypothesis at the start. So like I actually I didn't plan this analysis. So like, oh yeah, if I break the analysis and just look at this subgroup, look, the effect seems like really real. It could be interesting, right? It could be real. But of course, if I know you tried a lot of different subgroup analysis until you got into this and okay, this really worked, then I know that maybe, I mean, it could be interesting, but you're generating a hypothesis, not testing, right? So you look at the data, you saw this pattern, but of course you can't do a statistical test and to confirm the hypothesis, the data is self-generated, right? That's circular. So
you're using the data twice.
If that wasn't planned from the start. It counts as okay. This might be interesting, but it counts as an observation, not as a proof of anything, right?
Okay, no, makes sense. Now I would like to try to understand what the hell we can do with this situation and trying to fix it in a way. And indeed, I think it's interesting to start from what you said, that the clinical research is very different from the fundamental research. And I think also as a short clip that shows how much different can it make if you look at the same data from these two different points of view.
There's a lovely paper that Eric Turner and his colleagues did in the New England Journal where they went and they looked at clinical trials of antidepressants. And if you look through the journals, like you or I would if we went to the library, this is what we'd find. We'd find a large number of trials up here that show that these antidepressants work, and a very small number of trials, 3, showing that they do not work. Eric was working at the FDA at the time, and so Eric had a different view. Eric was able to see this. Eric was able to see, well, you've got a bunch that show that it works, but you've got a ton that show that it doesn't work.
So basically the difference there was that if you go for a clinical trial, you have to show all the data. So he was able to see everything that didn't work.
You have to show the data to the FDA, right?
To the FDA, yes.
Not necessarily to the public. So like if you're not at the FDA, well, there's the freedom of information, you can probably require it from the FDA, right? But there's all kinds of like legal things. I mean, the FDA may cross. So it's not like everybody has that view, right? The view of the average doctor is just a view from the upper part of the slide, and that's a big problem. That's what I mean. I think clinical research has had progress in that sense. In that, pre-registering a trial these days is actually a legal requirement for clinical trials in many countries, including the US or Brazil or I don't know the specific laws in every country, but I think it has become standard practice for clinical trials. It hasn't solved publication bias. Like there's still stuff that goes registered that is never published, and that's actually a relatively high percentage. But at least you know what I mean. That fixes both. One, you know what goes unpublished. So like there was this trial. It is started. It's somewhere. So like if it's missing, I know it's missing, and I can I can just say, okay, oh, see, there's a missing trial here. It could be better. I mean, there should be fines for people who do not publish trials. They were never applied by the FDA, but I mean, so there's room for improvement. But the other thing that pre-registration addresses, and I think that's interesting, is like to pre-specify your outcomes and your analysis strategy. So like that really limits. I mean, doesn't limit. I mean, you can still. I mean, and people do change their outcomes. They do change their analysis options. But again, you are able to see that, right? So you know, like, if they were actually looking at death, but in the end they reported percentage of people with chest pain, you know that like actually the primary outcome was negative here, and they looked for something else. And of course, it could be that it has an effect on this, but if you know they tried 3 or 4 or 5 things until getting into something that works, I know that this might not be as trusted as. So I think pre-registration is a great idea. I think it has made strides in other areas as well. Psychology has a big push for registry reports, right? So like, which is a slightly different. Not only you register your protocol, but actually submit it to peer review. So like peer review happens at the protocol stage.
So before the publication, before starting the research, you said,
doing
this is what I'm gonna do.
And then you get like an in-principle acceptance. You've actually do the experiment. In principle, you get published no matter what the results are. Again, I think that publication bias, it addresses analysis flexibility. That said, it doesn't necessarily work for everything, right? I mean, that works for like studies that are very clearly designed from the beginning, which is the case in clinical trials. You should have a very specific protocol, and that's followed over months or years sometimes until you get to a final result. If you go to bench science, I mean, sometimes just like do an experiment, see what happens, then this actually leads you to think of the other one, and there's a you can really pre-specify everything from the start. So I don't think this is a one-size-fits-all solution. I think it's great when you can pre-register, but that only works from actually you have a very clear idea of what you want to do from the start. So it's really like in the confirmatory science field. Like I know what the hypothesis is, I know how to measure this, I know how to do experiments, I know what and then I think pre-registration is a great step in terms of okay, you can actually see my protocol before, and you can see that I follow this, and this is all. I'm actually testing a pre-specified hypothesis in the way I plan to, and then statistics actually make sense and gives you information. That said, again, I don't think that stuff comes up, right? I mean, sometimes you just plan to measure this way, and you can never get this technique to work, and then you have to. So, like, you can do this for everything. I think you can do this at the level of the individual experiments for much of bench science, but not for a whole project. So, like a whole paper, like 30 experiments in it, there's no way you can pre-register all 30 at the start, because I mean, you change paths, and you don't want to kill exploratory research. So, I don't think you can make like register reports like the default for everything, and I think that's one thing to be. But when you do have a very very clear protocol from the start, I think pre-registration is great. And even like in more exploratory research, you can still like pre-register individual experiments, right? Like the protocol comes up here. Not sure who's gonna check it, but like you can actually write out the protocol and make it public before doing the experiment. Even if it's a short summary, I think that's a long way in terms of transparency, right?
Okay. And going a little bit in another direction with this question from Melissa, how much the reproducibility crisis is related to research fundings, because we know that, of course, if you publish, you get the money. If you get the money, you do more research. Do we know if, for example, I think I saw some research in which they were saying that maybe non-reproducible data are more cited, even though I don't know how much is easy to understand, or is there an issue there in that sense?
Yeah, that's there is that data in the I mean, people have done that analysis in the reproducibility psychology, and in the one of the Amgen paper as well. But I think those are small differences and small samples, right? So I would go that far as say this is like something we know is a fact for all of science. That said, I mean, of course, I think funding is very much a part of it, but I think in the end, one problem is just like funding has become very competitive, right? So it's hard to get a grant, and I think we actually measure people from. I mean, one thing I think is also important. Not only funding realities are vastly, like what people actually look at are vastly different among countries. So like there's a first road, very high impact science view. But I mean, if you're in Brazil or Africa, or I mean, people might look at very different indicators.
Okay.
Like in say first road top research university science, I mean, people are probably looking at like very high impact papers in terms of like Nature, Science, Cell, whatever. And those have the problem with those journals, I mean, nothing against the journal, but like to make it into a science paper doesn't, I mean, just being methodologically solid is not enough. You need to have like an exciting finding, a new discovery, something that makes an interesting story, right? And of course, if career advancement depends on publishing on those journals, in which like just being a good scientist is not enough. You actually need to like be lucky enough to like, get really exciting results. And most projects won't really give you most stuff just is boring or doesn't work. So like. You can't judge people on the basis of the results, right? I mean, that's I think like, and that's a very you're taught from the beginning. Okay, you shouldn't mess with the results. At the same time, you know you're being like watched and financed, and your career depends on them. So like, it creates a very perverse incentive. I mean, you can't. I mean, you can make career advancement depend on results. I think that's fundamentally wrong, right?
Wrong.
And I think like again placing very much of the revelation like very high impact publications in top journals that will require exciting results for you to make that's a recipe for disasters. In other places, I think like in Brazil still, I think it's a lot of it's about just like numbers of publications. It had like, I think it's getting better, but like we had like from the 90s to the 2010, like for 20 or 30 years, like people were very much judging like the number of, so like it's like I published 50 papers in this year, and that's also stupid. I mean, right? So
doesn't mean that it's good. Yes.
Because the best way to like publish a lot of time is just like to be less rigorous and not do like a lot of quality control. Like the more rigorous science takes more effort. So like in the end, I think you have to reward people for rigor more than for more than for numbers or for impact, right? I think both numbers and impact have a problem. Like the central dimension of science actually getting it right, and I think it's like not rewarded as efficiently, perhaps because you don't really have simple metrics for that, right? I mean, you can count citations, you can count papers, but it's harder to count rigors. I mean, it's not impossible, right? I mean, you can agree on transparency metrics. Did you share your data? Did you share your code? Did the statistics look right? Did you blind your experiment? So I mean, there's of course, I mean, whenever you make a metric a goal, it seems to be good metrics, so like people can just like start cheating the system. If you just like hey, it has to be blinded, everybody will say they blind the experiment, right? But I think there's you can try to make a call on like how transparent people are. I think that's possible to judge, and actually that might be easier to judge if people are transparent than like their work has a big impact or is very exciting. I mean, but we do have proxies for the latter, right? And like, so we can use the impact factor for the journal, and like that's a general measure of like how interesting the finding is, which is a bad measure, but I mean, there's a proxy for that. I don't think we have developed as well, but there's something. It's possible to have metrics for rigor and transparency, right? I think we just have to make them easier to collect and agree on them and getting them to be part of evaluation, right?
Yeah. And there is also an interesting question. It's something that we skipped a little bit before on why we have this issue. And Annika she's saying that there is also a problem of the high competition. So as you said, funding is difficult to get. So we need to publish, and you need to be the first to publish. And I'm interested in this because I always think, and I know it sounds crazy to say it out loud, but I always think about Galileo. You know, he was maybe the first to use the scientific method, and he also published something that maybe he was not totally sure, and he had a very hard peer review, which was the Inquisition. But still, it's something that we do it basically from the beginning. We want to be the first one to publish.
Yeah.
But how do we find a way to make these two worlds collide decently?
I think, and a lot of novelty is just overrated in science. I mean, you can be the first to publish. I mean, it's not that it's, of course, like thinking of something that nobody else has. I think it's interesting. But again, like if you have like a very small study that suggests something, and then a very big study that like really confirms this. For first has 30 patients, and the next one has 3,000. In the end, the 3,000 study is what really matters, right? So like. I think doing it well and doing it rigorously, it's more important in the end for the scientific record than doing it first. Of course, that doesn't hold like ideas or theory. I mean, then we can discuss it. Like, but like in terms of like just like generating data, it shouldn't be a big problem if you come second, right? If you're actually generating much more data than the first one, that's still a contribution. And I think a lot of the failure in biomedical science is just like not being very rigorous in confirming stuff. Like somebody publishes first, and this seems like an exciting fact, and that's a very small study. But like then there's like somebody else finds it in a small study because of course you have like confirmation bias, and you're kind of trying to get the same result because the big shot there got this. But you have like this. I don't know, in the field I work in, like in the neurobiological memory, have like this 10 or 20 studies, but like each one has like 10 rats in each group, and it never goes up. I mean, like, if we're gonna make this into like a consensus and dogma, let's try to get like 4 or 5 labs together, like do it in like 100 mice, and then okay, we all agree on the methods. It's all pre-registered. Okay, boom. So we can confirm this in other places. Okay, now we move on. But like I think, I think the obsession for novelty makes like people to publish when they're not necessarily sure because they want to be fast. And as there's not much incentive in terms of like confirming a finding, nobody really. I mean, people do replicate eventually, just like to start their paper. Okay, I got the same thing as that guy, but I mean, there's little incentive for like really. Okay, this sounds interesting, but like let's get a lot of people involved and like make sure that this is actually reproducible in a lot of places. I mean, there's those grants are not very common or non-existent in some places. And again, I think clinical medicine does it better. I think like if you open up the big medical journals, there's a lot of stuff like, okay, this has been popping up in the literature now and then, but like now we did the 5,000 patient study and this. So I think there's a little bit more. It's not that novelty doesn't count. So like it's not that they completely solve the problem, but I think there's more, especially as there's more tradition of data synthesis. I mean, you do have much more like meta-analysis, and of course, if you do meta-analysis, the 5,000 patient studies counts 100 times more than the 50 patient one, right? So, so in the end, what counts in the guidelines is the larger studies. So I think
I understand.
I think people do value more that okay, this is a very large study, gives you more points than in the basic science.
Okay, no, it makes sense. I mean, we should reward more the good one rather than the sexy one, I would say.
Yeah, or maybe just a large one sometimes. I mean, just like
the larger one. Yes.
How much data you collected in, right?
Yep. And to go in that direction, so if we want to go maybe towards the clinical trials, I mean, probably the asset of research that we have right now is a little bit hard to keep using because we don't have these big centralized systems. And I mean, how can we go there? I mean, do you envision a system where we have huge research institute that only do that, or maybe national institute that do that, or consortium? What do we go?
I do envision that future. I can't make it happen because it's hard. But I do think that consortia and big collaborations and eventually big research. I mean, I think you can get it from like all the sources. That said, I think the first thing, okay, we do need this. I think that's the message at first. I mean, there's been interesting. We run a large collaboration, which is the Brazilian Reproducibility Initiative. There's like a 60-something labs strong, and we're replicating. It's a replication project, but we're replicating things in like a lot of labs across the country. There have been interesting grassroots project like the Psychological Science Accelerator, which is like I don't know 300 something labs strong around the planet. They have this government system. Okay, let's try to see what's an interesting problem, and if we do have one, then we get like 100 labs to work on it. There's many babies, many problems. There's all kinds of interesting collaborative projects popping up. That said, I mean, those are hard to run. It's not always easy to get funding for that, and they run in a rather amateur way in terms of like this, just like regular scientists which have their day jobs doing something else. They're like, okay, we're interested in this, but it's just like your normal academic scientists trying to morph into something more confirmatory and more professional or more whatever you call it. That's not always easy. I mean, people are not very used to having centralized. I mean, again, clinical trials work because they have been largely taken by industry, which has a very professional way of running them, like very standardized. They hire somebody who does specifically recruits patients, somebody who specifically. So like it became a much more specialized kind of work, in which managing is like a very big part of it. So like if you actually look at what big pharma companies does, they actually manage people, because like clinical research is being done by some biotech startup. The clinical trials are being done by CRO. The hypothesis and analysis come from like people from academia, which is hard. So like their business is actually like getting people together and putting money and doing the. And I don't think we do have a structure for that in most of science. We're very atomized. So like if you just like distribute money and you organize a system in like this little labs which are 5, 10, 15 people strong, getting them together in an organized fashion to like do is actually very hard. Our experience in running this kind of collaborations, like people are just not used to being told what to do, and that's perfectly fine. I mean, it's not perfect. But again, as a centralized project, we have to like standardize stuff, and so it's hard to sometimes it's hard to getting people to like answer emails. Like it's not like I'm not their boss, right? So like to actually get everybody together, like and run it smoothly can be a huge challenge, because people have other priorities. They mostly doing this as a, and I don't think like if they think it's important, but like some are working on weekends. So I feel we're this, I don't know, the circus crew sometimes that of playing people are just like not professional enough to do it. And I'm the leader.
The captain.
I mean, sincerely, yeah, but it's like Brancaleone's army, right? It's not. I think we need to build the structures, and I think this can arise from like multiple ways. You could have this like very large institute that like, okay, we do replication in a very and we have this very controlled process. That said, I think you do lose some. I mean, if you just like do it in one place, even if it's a very high quality place, you lose the kind of like heterogeneity that exists in the world. I do like better the idea like, okay, we get this done in multiple places. And we do have a lot of labs to do stuff. I mean, but I think like coordinating and coordinating in a way that is not purely like what we normally do as academic collaboration, coordinating with like more specialization of labor, like somebody just like coordinates this, somebody just analyzes this, somebody collects data and gets rewarded for collecting data. I think we have to move in that direction. Like not everybody in science has to run their own lab and be a PI and have their own results. I mean, I think we could have much more people actually engaging in this collaboration. Because, okay, I professionally do experiments, I collect data, and I get rewarded for this. I think, and some people should be actually, we need people to run this actually from in a specialized manner, not like I'm getting 60 labs together and trying to figure out how to do this, because I mean, managing is a full-time job.
Yes.
So I think you do, and I think, and we do need people to actually pick the questions which we actually need this stuff to plan. I mean, it's not like you can't do everything in 50 labs. I mean, we have to like, okay, we have to pick what's really important in this field, what we want to confirm, what we have doubts about. So, okay, like this and this and this problem this year, we'll do this 50 lab experiment just like to very accurately measure this effect, because this is vital for the field. This is important to build theory, etc. So, like, I think selecting what deserves to be confirmed is actually a very hard task. There needs a lot of specialized input in terms of like people who actually work in the area. So, I think I'm not sure who should do this, like scientific society, but I think communities in a specific topic need to
discuss it.
Yeah, discuss this. Like, have a formal way to like decide what we should test. And then we need like a structure to test a few hypotheses. It's not like we need to do everything this way, but like we need to be able to do the large experiment when we want to. And I think we don't really have that structure, and building it from scratch is hard. like having like everybody who wants to do a big lab collaboration to learn how to manage this from scratch is not very efficient. So like if you actually have a more permanent structure of like people who can get the community's input in terms of like what are the important questions, know who to call. But so like I think it's like a science managing thing that might be run by funding agencies or might be run by academics who do that full time. I think there's different ways to do it. I think some are more collaborative, some are more centralized. I think both can work, right? I think,
I agree. But I feel it's something that we could speak for out of 6 hours, and unfortunately, a little bit running out of time. But I would like to close with one question, because indeed, you're running this big project with the Brazilian Reproducibility Initiative, and what was so far the most surprising thing that you found that maybe you were not expecting when you started this journey?
A lot of labs joined. So like,
good.
No, seriously, we didn't expect to be so large. I mean, when we started out the project, we thought like maybe I don't know, like we did a lot of outreach in terms of like doing lectures and using social media and like trying to get people to join the project. That's like our goal was like 30 to 50 labs joining. 50 was like very optimistic. And I think at our first call, I think we had like 71 labs signing up.
Wow.
We had some extra calls just for specific stuff. I think in total, 90 something labs eventually like filled up a form. Okay, I'm interested in joining the project. I think of those, we managed to get maybe 70 something in the project eventually. Some left because the pandemic really hurt us in terms of like a lot of labs closed down, and when they opened up again, the people had moved, and so they had to leave because like we had like something close to like 10 to 15 labs actually like leaving. We have 60 something at the moment, but that's definitely much more than I expected. So like, and that's within one country, right? Within very specific mix. So like people are interested. I think that's the optimistic view. Is that people are interested in this, and there's enough people interested in this to actually make collaborations happen. The downside is like it's very hard to get all these people together in an efficient fashion. So like I think the bad surprise is like I didn't expect it to be easy, right? But I think we do stumble into like simple things like simple communication problems or like. There's a lot of bureaucracy that could be made easier if we had like a more professional system. Brazil has its problems in terms of like getting supplies is very hard. So like you have to import a lot of stuff, and that goes through like all kinds of bureaucracy. There's special licenses. We spend like ridiculous amounts of hours trying to get licenses for specific products. Sometimes we didn't get them in the end. So like we're cutting experiments because we just can't buy this. So like. A lot of our work is actually could be done easier just by like one, simplifying bureaucracy, which in Brazil is a big problem. Two, making just like having specialized people do specialized work. So like buying reagents by itself, we actually these days we actually employ a startup to just like buy stuff for us. Okay. They know where to find, and that makes a lot of sense. Like to pay people to actually do what they do, and then if they know how to do it, right? But again, grants are not usually awarded for you to like be able to hire a manager like a big project. So we need to realize that contributions other than just like doing experiments around it's actually important, and we need to reward people who like work into the structure in science and get them. I mean, we do have full-time people in universities and in labs that actually do full-time jobs in terms of like organizing stuff. I think we just need to bring these people into this large collaboration. I think some fields have done better than others, right? I think if you look at genomics, for example, I think they did manage to build big consortia which were not there like 20 years ago. I think clinical medicine does run large clinical trials, although that has been now largely outsourced to industry. But I think, and physics, you mentioned that's, you have like a CERN thing that can test things that like nobody could test by themselves. But you have this big collaborations in astrophysics and particle physics that are thousands of scientists strong, and each one doing their own. And then they sign the papers like sometimes like 1,000 people in alphabetical order, and that's well accepted. So I think we need to move into a research culture. Like big science is needed sometimes, not always. I think we need the small teams as well, but like sometimes. And we need to have a professional, organized, long-term structure for this to happen when it needs to happen, right? And I think there's work to be done in that sense.
Of course. Well, for sure, I for one, thank you for this job. I mean, I know that it's a huge undertaking, and it's great that there is someone that is willing to do it. And I think, and also our viewers the same. I would really like to stay and speak for other hours because it's super interesting, but I'm afraid that our time is limited. So I thank you very much for being with us, Olavo. Maybe we're going to have a possibility to speak again in the future. And I thank everybody for watching. So we are not going to be back in 2 weeks. We take a skip. So we're going to be back on the 13th of April. Of June. Oh, sorry. We are going to be back on the 13th of April. So stay tuned with our social media because we will be back. And in the meanwhile, you can check all our other activities on academy@tox.pe. I hope you had fun. I definitely had. And thank you very much for watching and see you next time. Bye-bye.
Bye.